Good day, everyone.
My name is Nur Najwa Aliani from University Teknologi Mara, Perlis.
Today, I'll be presenting our work on molecular insights into the dinuclear nickel site of Jack-Bean urease. Urease, also known as EC3.5.1.5, is a nickel-dependent metalloenzyme. Its role is to catalyze the breakdown of urea into ammonia and carbon dioxide. Why is this important? In agriculture, urease controls the efficiency of urea fertilizers. In medicine, urease-producing bacteria such as Helicobacter pylori and Klebsiella pneumoniae are linked to gastric diseases and urinary tract infections. In the environment, urease activity is central to the nitrogen cycle. Despite many studies on urease, most have focused on docking or mechanistic pathways. What remains less understood is the electronic properties of the nickel site, and especially how the surrounding residues influence it. Previous research tells us how substrates or inhibitors bind, but less attention has been given to the electronic environment around the nickel atoms. This is important because the nearby residues may strongly influence reactivity, molecular recognition, and catalysis. That's where our study comes in. Our objectives are threefold: to investigate the molecular and electronic properties of the Jack-Bean urease active site, to compare truncated models, 6 residue versus 12 residue clusters, surrounding the nickel atoms, to benchmark computational methods, density functional theory, or DFT, at the B3LYP/6-31G level with dispersion corrections, against the more computationally efficient semi-empirical XTB method. Here's our workflow. We first extracted the active site residues from the protein structure. 2 models were made, one with 6 residues and one with 12 residues. Both models were optimized using GFN2, XTP for efficiency. Final calculations were done at the DFT level, including dispersion corrections. Post-processing analyses included electrostatic potential mapping and non-covalent interaction analysis using tools like Multune and VMD. Let's look at the results. For the nickel-nickel distance. In the 6 residue cluster, the distance expanded from 3.675 to 4.035 angstroms. In the 12 residue cluster, it only expanded slightly to 3.969 angstroms. This shows that including more residues preserves a geometry closer to the native structure, whereas smaller clusters exaggerate flexibility and destabilization. Electrostatic potential, or ESP, surfaces tell us about charge distribution. The 6 residue cluster shows large imbalance, regions of both extreme positive and negative charges. In contrast, the 12 residue cluster displays a more uniform surface, reflecting a balanced electrostatic environment. Quantitatively, the 12 residue model also showed deeper minima and higher maxima, indicating stronger polarization that is necessary for substrate recognition and inhibitor binding. Next, the noncovalent interaction, or NCI, analysis. Green regions represent van der Waals forces. Blue indicates attractive interactions, like hydrogen bonding or coordination. Red signals steric repulsion. In the 12 residue model, we see stronger attractive interactions near the nickel atoms, which help anchor the metals in place. Interestingly, steric repulsion also plays a functional role, restricting flexibility and defining the boundaries of the active site pocket. We also tested how urea interacts with the active site. Results show that nickel-115 interacts more strongly with urea than nickel-116. Additionally, residues such as histidine, lysine, and aspartate were observed to assist in binding. This points to an asymmetry between the 2 nickel centers, which could be important for inhibitor design. To summarize, the 12 residue cluster provides a more faithful and realistic representation of the urease active site. It preserves geometry, captures stronger polarization, and maintains a richer network of noncovalent interactions. Functionally, nickel-115 plays a stronger role in urea binding than nickel-116, showing asymmetry within the dinuclear site. These insights not only improve our understanding of urease, but also suggest strategies for designing selective inhibitors. We would like to thank the Ministry of Higher Education Malaysia for funding this project through the FRGS grant. And thank you all for your attention. I'll be happy to take any questions.